Chromosomes of human cells have a structure called telomere, which caps their terminal regions. The significance of this is in regards to cellular division. After each division, the somatic and stem cell telomeres gradually become shorter. The top image on the right showcases the process of DNA replication where chromosomes shorten. On the bottom of this image, we can see that once the RNA primer is removed, the single-stranded DNA sequence is degraded, and thus the chromosome shortens. And this, of course, on both ends of the chromosome where there are telomeres present. So, how do cells remedy this? They utilize what is called telomerase, which replicates telomeres using an RNA template. The bottom image here, all right, helps to show a kind of visual of how telomerase operates in extending the repeated DNA sequence in the 5' to 3' direction. By extending the sequence, telomerase makes room for RNA prime to be added, allowing for additional repeats in the extended strand and thus preventing shortening of telomeres during replication. So now that we've gone over some of the basic ideas of telomerase, one might ask just how all of this ties into cancer treatment. Well, as we previously discussed, healthy cells can only divide a certain amount of times until telomeres become so short that reproduction is halted. So while telomerase activity can elongate the presence of certain cells, somatic cells lack this activity. On the contrary, the vast majority of cancerous tumors activate telomerase. Most cancer cells express this high telomerase level, while others maintain their immortality using alternative lengthening of telomere system, or ALT for short. Due to these observations, the inhibition of telomerase has become a hotly debated topic on the basis of cancer. In some sort, if some sort of specialized treatment could halt the expression and or production of telomerase in cancer cells, then theoretically, like human somatic cells, division would only occur a set number of times before reproduction is halted. In terms of and this course, telomeres are quite useful. So firstly, certain assays and tests involving telomeres are relatively inexpensive and are often quite safe for laboratory use and handleability by students. So they get a lot of hands-on experience. They operate under different scenarios. And additionally, students may benefit from learning about these different scenarios because telomeres aiding in DNA replication, they're very useful in terms of studies in aging, sequencing, transcription, translation, just to list a few. So in terms of this course specifically, in terms of enzymes, in terms of proteins, in terms of application with disease treatments and the laboratory, there are tons of applications for which students can find extreme. And I personally think it is an incredibly important topic that could open the doors for a lot more studies in terms of treating diseases and other processes.